Welcome, and thank you for attending Pharma Collaboration to Advance Microphysiological Systems, a presentation from the iqmps affiliate. My name is Szczepan Baran, and I'm a head of emerging technologies within comparative medicine at Novartis Institutes for Biomedical Research. However, today I'm presenting as a vice chair of the iqmps affiliate. I would also like to thank the conference organizers from the Laboratory Animal Sciences LabRoots conference for establishing this track addressing MPS and also giving us an opportunity to present an iqmps affiliate perspective. Before we jump into different aspects of MPS, it is imperative for us to align or at least to be on the same page. What do we define or what do we mean by MPS? So the iqmps affiliate defines MPS as going beyond traditional 2D culture models, where we include several of the following design aspects: multicellular environment within biopolymer or tissue-derived matrix, a 3D structure, an inclusion of mechanical cues such as stretch or perfusion for breathing, gut paralysis, flow, and incorporating primary or stem cell derived cells, and or inclusion of immune system components. Below you see an image that helps us visualize the definition, which was adopted from Dash and Proctor. So this is a pretty broad definition. As you will see as we move through the presentation, depending on the context of use, this definition can be refined. But this is what we define as MPS by iqmps affiliate. So as many of us know, the microphysiological systems have a high transformational potential. The first context of use commonly explored with these technologies includes predictive and mechanistic safety. More recently, we have been looking from efficacy and disease modeling focus with broad applications and entry points. The level of qualification that's required for implementation is really dependent on the area where MPS is employed, depending on the context of use, which you will hear me describe that today or talk about that today, but also most people whose presentations you will hear will address that as well. And of course, pulling all of that together, these technologies have enormous potential to reduce, refine, and hopefully in the future, replace the use of animals in biomedical research. So where do these technologies fit in? And really, we need to think from that perspective of what are our challenges, what are our problems. So for example, where don't we have in vivo models or good in vivo models? So the exciting part about MPS is they can fit into any stage of the drug discovery and development journey. So in target discovery, we can look at also target safety assessments, target validation. For early discovery, help us optimize our leads. And then even moving into the early clinical development. So addressing or helping us mitigate some of the issues that come up there. So the point of this figure is really that depending on the context of use, this technology can potentially fit in and help us address some of the challenges that we are facing. So how do we engage? What's the best way to do that with MPS technologies? So as we know, again, there's a high potential for these technologies to be transformative. And with any novel or emerging technologies, this time to show the impact of these technologies can be prolonged or very long. So what's the best way to kind of engage at a faster pace versus the traditional or the current approach? So as an affiliate, we have come together and identify the various areas which would allow us to expedite this path. So one, partnership or collaboration. This is imperative. So working with all of the stakeholders, industry, academics, government, regulators, and any other stakeholder that's involved in these technologies. So looking at the performance and capability development, what's really needed. Then defining the context of use, right? Where we're going to use these technologies, where it's going to fit for us. Is it a mechanistic assay, safety screening, DMPK, disposition, efficacy? And then, of course, characterization. So what is the cell or tissue composition of these technologies? What's the physiological function? So having an understanding there is going to help us as well. And then looking at validation, throughput capability, interlaboratory reproducibility, the stability of these platforms, and what is the effectiveness. And there can be different definition of characterization versus validation. There certainly can be some overlap there. And then the fifth aspect is commercialization. So what is the data that we would need as end users to engage and incorporate these into our daily processes? And the data that would help us with our decision-making process or provide actionable insight. And this is pulling all of this together, right? I think in some times the technology providers believe that, hey, we're there, this technology should be utilized, everybody should be using it. And as an end user, well, maybe we're not there yet. We still need this, or that. So really pulling this information together and bridging that gap between the technology providers and the end users, working collaboratively, we can certainly help engage and expedite engagement with these technologies where everybody benefits. So how did iqmps affiliate come about? So first, let me describe the IQ. So IQ, which is International Consortium for Innovation and Quality in Pharmaceutical Development. And the IQ, which is formed by, I believe, 38 or so pharmaceutical companies, has leadership groups, which you can see listed on the left-hand side. Each leadership group also has working groups or specific working streams that address specific topics. So in 2014, within the 3Rs leadership group, an MPS working group was established and involved 11 companies. And one of the reasons for establishing this is that the National Center for Advancing Translational Sciences, or NCATS, wanted to learn where does our industry see these technologies. So in 2015, we signed an MOU with NCATS. In 2016, the group actually presented at the NCATS Tissue Chip Workshops and 3D Tissue Model Summit. In 2017, the group published a position paper, which I just showed a couple slides before, on these technologies, how to navigate chips from development to dissemination to end user utilization. We also have developed poster presentation on 5 sets of organotypic standards, which served later on as a basis for our manuscripts. And in 2017, we also hosted a face-to-face workshop on industrialization strategy. So we really wanted to figure out where should we go? What are our objectives? What are our goals? So we got into a room in DC, and actually at that stage, we were still, again, not sure exactly what we wanted to do. Everybody had different ideas. So within, I think 7 or 8 hours, we were able to do that. And what we recognized that being a working group wasn't enough for us. Our aspirations were really high. There was a lot of projects that and objectives we would like to reach. So that's where we decided to establish a microphysiological affiliate. So currently, this affiliate is made up of 21 companies, as you can see listed here. The representation is very diverse, from drug pharmacology, safety, 3Rs, ADME, PK, PD. And that has certainly evolved. The meeting that I mentioned, face-to-face meeting in 2017, I think out of the 12 or 13 or 14 people in a room, I was the only non-toxicologist person in that room. So obviously you can see that the representation has diverse since then. So our current leadership, our fearless leader is Jason Eckert from GSK. I have the honor of being the vice chair. Vice Chair-elect is Rhiannon Hardwick from Theravance. An immediate PES Chair is Terry Van Vliet. And a PES Chair is William Proctor from Genentech. So iqmps affiliate has really 4 aims, overarching aims. One is for us to serve as a thought leader for these technologies for developers and stakeholder organizations. Also provide a venue for appropriate cross-pharma collaboration. This is where also data sharing aspect comes into play, and I'll talk about that in a little bit. And what's the best way for us to do that? And also engage with the regulators. So when emerging technologies, innovative technologies come about, very often we work with them, we collect data with those technologies, and then when we think we're ready, this is when we engage with the regulators. And obviously that can prolong that process of engaging or deciding where to implement those technologies. So here we wanted to engage with regulators at the early stages. So from the beginning, we can work collaboratively and help evolve these technologies in this field. We also have an external partnerships and collaboration goal. And as we know in this field, there's a lot of other organizations, consortia, or groups that are engaged with these technologies. And we want to make sure that we don't duplicate efforts that are happening somewhere else, or identify opportunities where we can collaborate to actually help move this field forward. So our current work streams, we have 5 of them. One is organic manuscripts. Then we also have project proposal teams. We have a gap survey team, regulatory outreach work stream, and the communication external organization outreach work stream. And I'll talk about each one of these in more detail. So the landscape and gap assessment survey team, the goal for this team was to really understand how pharmaceutical companies are engaging with these technologies. How are we using these technologies? And when we think about these complex in vitro models, and what are some of the challenges? Why aren't we engaging with them? And how can we realize full potential of MPS? So in 2019, we conducted a survey, and then in 2020, we actually redesigned the survey, also working with FDA, incorporating FDA feedback to the survey 2.0. This survey was launched at the end of last year, and we have collected the responses from it in early January, and now we're processing and analyzing the data. And the objective for this group is also pool the results from both of the surveys and develop a commentary manuscript where we're also going to address longitudinal analyses of both of these surveys and the results from these surveys. So for our first survey, the definition that we used for complex in vitro models, or MPS, is pretty broad. So this is the definition that I gave a few slides earlier, so I'm not going to really go into that. And we really wanted to have it broad so we could engage as many stakeholders as we could. They have been working with these technologies or are even just interested in these technologies. Our 2.0 survey, we have refined or refocused the definition, and you can see that on the image there at the bottom of the slide. So our initial survey included all 5 of these aspects, and for our 2.0 survey, we end up including that last 2 aspects. So here we define in vitro systems that could include 3D bioprinted tissues, microfluidic systems, or combinations thereof, with fluid or airflow, coupled with cells, and these could be primary cell lines, and stem cells, and they need to recapitulate components of organ physiology. And in this survey, MPS does not include static organoids, spheroids, or micropattern conculture systems. So I'm going to go through some of the questions that were presented within Survey 1.0 and the answers to those questions. So first, we wanted to find out, who responded to the survey. What is the makeup of the folks that participated in there? So you had 28 respondents. You had 23 members of the iqmps affiliate that participated. We actually had 5 additional companies that were not members of the iqmps affiliate, but did participate in the survey. The survey was 97 questions, right? So it's not one of those 5-6 question surveys. It was pretty intense. It took significant amount of time and effort to complete. So if you think about the number of respondents that we actually received was really high, it just demonstrates the interest and then engagement of these pharmaceutical companies in these technologies. So then we subdivided respondents into small, mid-size, and large. So for small, it's less than 1,000 employees, mid-size, 1,000 to 10,000, and large, over 10,000. And as you can see, majority of the respondents were large companies, so 18 of them. And then for mid-size, we had 8, and for small size, we had 2 companies. So the question also was, so have you engaged with these technologies? So 26 companies said yes, and 2 companies said no. That said, though, even though they said no, they are still interested in working with these technologies potentially. So the reason for not engaging with them was lack of resources, but also that the key decision makers wanted to see where this technology, wanted to engage with the technology after it already matures a little bit more. So what's the rationale and advantages of engaging or using these technologies? So the major 2 were lack of current tools or systems, and improve current models' ability to replicate human and or animal physiology. And third, improve current assay capabilities was right there as well. And then other, which included 3 R's, motivation, mechanisms, translational toxicology, and human biology. So what are some of the observations that these organizations have made that were advantageous with engagement? So the largest percentage was 22 companies, replicate physiology more accurately. Then we had more sensitive systems. They capture more endpoints, capture more time points. And then other also included, longer duration of the system of the cells being alive, live imaging, where it's not possible, for example, with in vivo models. So how do you resource these technologies? So this kind of was a pretty even split, right? So almost 90% resourced it in-house. A little bit over 80% was through external collaboration. And then almost 80% was fee-for-service. And then there was a smaller percentage, which were other, for example, academic collaborations. So where does each company source these models from? So the highest number was from vendors, so commercial products, and it makes sense, right? It's easier to engage with those technologies. You can just jump in, bring it in, or potentially use it, or use it as a fee for service. Then we had academia, which again, these are early stages. This field is in early stages, so it makes sense to engage with academic groups that are evolving, developing these technologies. Then we also had vendors and internal as well. So some companies are developing these systems internally. So how internally we are addressing resources for these technologies. So how many people are part of engagement or internal efforts with these systems? So 40% or almost 40% had anywhere between 11 to 50 people. And then we had 5 to 10. And then over 50%, only a couple companies had over 50 associates engaged with these technologies. And for us, obviously, for all of these questions, but specifically this one, will be interesting to see how that progresses from Survey 1.0 to Survey 2.0, especially when we refocused the definition of MPS. So then, what divisions, what departments are actually working with these technologies? So again, there was almost like a three-even split. So you had DMPK, research biology, preclinical safety, safety assessment. We also had biomarker discovery and some other areas like disease areas, discovery therapy, early clinical development. And one of the points to take home from here is the initial slide that I show that these technologies can really fit in almost anywhere from early discovery to later development of our medicines. One of the questions was also, are these centralized efforts, right, or not? Are these segmented? And as we know, sometimes it can be challenging when things are siloed. So we really wanted to gauge that as well. And only small percentage of companies responded, only 2, that those efforts were centralized. Not surprisingly, within most companies, these efforts were segmented. There was a few companies that actually had other ways. So had a core or a mix of both, of various working groups. So of course, it's always interesting, right, or good to learn where our decision makers or key decision makers are where are they thinking? What are they thinking about these technologies? Could this have really big impact or not? So really high percentage, over 50%, believe that technology has value and great potential, and already are investing and are planning to invest and continue engagement with these technologies. About 30% said there's unproven to date, so there's more information needed regarding these technologies to demonstrate their value. But are investing in these technologies and willing to explore the potential of these technologies. And there are obviously some other reasons, like for example, is there a trust in the technology? But it's hard to manage the approval process. But no one really said that these technologies are not proven and unlikely to meet its potential, which is really exciting to see that everybody that participate in this survey sees some kind of potential and advantages to working with these technologies. Of course, understanding which system is of the highest priority is important, obviously, to us as an industry, but also for the technology providers, right? So where they can concentrate their efforts to engage with these technologies or develop these technologies. So not surprisingly, liver is one of them. Again, even just thinking back, right, when I said the first meeting that we had, there's mainly toxicologists present in a room. But obviously that has evolved, right? So then looking at GI, kidney, CNS, and immune system. So where are these technologies used, right? So I referenced the context of use, which is really imperative when we talk about these technologies. So on the left-hand side on the top, you can see some of the types of these technologies. And so here we have a chart, right? So in which area these technologies are used and the kind of percentage, right? So for safety, static 3D technologies are pretty common. You also have a 2D micropattern technologies as well. When you look at the ADME, again, the 2D micropattern technologies. And then for pharmacology, it's kind of evenly split, but the static 3D technologies, which again, not surprisingly. And so those are the most frequently used models. And one of the things for the second survey, where we again are focusing more on the more complex MPS, we're really curious to see if some of this data is going to change, and we really are expecting that it would change, but only the analysis of the survey results will tell us. But even here, you can see that engagement with the organ-on-a-chip technologies or the more complex systems, there is still some engagement. And you're looking at the mechanism, looking at the species differentiation, as well as screening in all three of these areas: safety, ADME, and pharmacology. But overwhelmingly, at least for this first survey, the most frequently used models were the 2D micropatterned and static 3D models. So at what stage of drug discovery and development journey are we engaging with these technologies? So preclinical safety, most commonly, then looking at lead optimization, and then it's kind of evenly split for target ID and validation and lead identification. And then obviously we do have some engagement also in the clinical development and in other areas like PK/PD, looking at the mechanistic aspect and drug-to-drug interactions. So what modalities are we testing with these technologies? So here overwhelmingly small molecules, but biotherapeutics are right behind. We're also engaging with cell and gene therapy. And I'd be curious to see actually with this new survey if that has increased, because this is certainly an area where we don't have a lot of in vivo models where we can test safety and efficacy of these therapies. And to a smaller percentage of engagement is in vaccines and other therapies. I'd also like to point out that commercial and active proprietary compounds and terminated proprietary compounds are equally used when evaluating these technologies. So, of course, a question that many of us are certainly interested in, has the data from these systems been part of regulatory submission, or where else are we utilizing this data to make decisions? So we are certainly looking at using this data to qualify models. It's certainly utilized internally for decision making, which makes sense, again, especially with emerging technologies. But 18% of the participants for the survey said that they have utilized this data for regulatory submission. And there are some other areas, about 30% or so, when you're looking at weighing of the evidence, looking at the mechanistics aspects, and also are planning, not currently did not, but are planning to include this data in regulatory submissions. So 25% they have submitted data to the regulatory agencies, which is 7 out of the 28 companies, stated that the data incorporated into non-clinical toxicology, human pharmacokinetics, and bioavailability, and non-clinical pharmacology sections of the regulatory filings. And the data was also submitted globally, so to FDA, EMEA, and PDMA. And 64% they have not submitted this data to regulatory agencies. So why is that? So they didn't have confidence in the data. There were also concerns regarding regulatory acceptance of the data in that submission. The models are not currently fully characterized, and also lack of confidence from internal stakeholders. And you saw some of the answers from the key stakeholders or decision makers, that some of them have thought that these technologies have not evolved yet or matured enough to potentially submit into these regulatory agencies. So to summarize, the outcome of the survey is that, which is I think pretty exciting, that these models are equally being applied in safety, ADME, and pharmacology. The resources are segmented across groups, which personally I hope that's going to evolve and we're going to see that in the second survey. The sources of these technologies are mainly from vendors, followed by academic, but also some internal development as well. The human and preclinical model species are equally being evaluated. There is some but limited interest in multi-organ integrated systems. And I think there, again, we really need to characterize and qualify these individual systems before there's excitement regarding the multi-organ integrated systems. For utilization of these technologies, currently they really are mainly used for internal decision-making processes. 9 companies that use these technologies use them for clinical development, but 5 companies also use them for data for regulatory submissions. So there's definitely progression and evolution where this data is being utilized. When you look at what modalities, so currently predominantly small molecule and some biotherapeutics, and there's definitely need for additional characterization and internal experiences with these models in order to increase the uptake and application of these technologies. These are definitely not, like a PCR kit that you can just grab from the shelf, read the instructions, and utilize. So having experiences with these technologies, setting them up, in addition to the other aspects that we kind of brought up or I have discussed and presented, and you will certainly see addressed in other presentations. But we're getting there. So now I'll switch gears a little bit and talk about an organic manuscripts working stream. So the goal for this team is really to summarize challenges and gaps in current in vitro and in vivo models, and what information would be meaningful to us from these systems to demonstrate the capabilities, and what is that improvement over our current standard models. And we also describe in these manuscripts, endpoints that are of interest to us. What are some of the readouts that we would like to see? And what compounds should be tested, right? So I mentioned earlier that, very often the technology providers develop these technologies, and we're not always jumping in, and engaging with them. So the idea behind these manuscripts is actually to provide some guidance on what we would expect from these technologies before we engage, or even while we're engaging, to increase that engagement and the impact of these technologies. So the deliverables for this group are pretty amazing. I mean, to be honest, since 2019, we have published 9 manuscripts. And just to think about, you have 21 pharmaceutical companies coming together and agreeing on even what endpoints to use can be very, very challenging, right? So to come together and really within less than a year or so to publish these manuscripts, to me, it was just an amazing deliverable. And we're also now looking at publishing or working on additional manuscripts. So as I already mentioned, that these manuscripts kind of evolved from the posters that were presented at the ANCET Tissue Chip Consortium meetings. And they really address the 3 questions that I kind of already mentioned, right? The challenges and gaps in current in vitro and in vivo models. And it's imperative to do that, to point those out. So then we can actually see what are some of the advantages and the meaningful improvement that these technologies would have over the other ones. And also, the endpoints, online readouts, test compounds, to share those with the technology developers. So on the right-hand side, you see manuscripts that were published, and on the left-hand side, you see the manuscripts that we're currently drafting. If you'd like to see these manuscripts, they have been published, they're open access. You can go to the link that's listed there, iqmps.org, and just click on a tab for publications. So here's the list of these publications. They were all published in Lab on a Chip. We have an introductory manuscript where we dive into a little bit more in depth, why we are publishing this series and the potential impact of these technologies. And then you have, the other systems organs addressing here, from lung all the way to cardiovascular. But again, you can access these manuscripts. They're open access on the website iqmps.org. So the next work stream that I would like to talk about is the regulatory engagement team. And I could go on and on about this work stream, and I'm slightly biased since I have the honor of chairing this group. But really the goal for us here moving forward is to continue our engagement with FDA. We're also drafting a manuscript from our workshop, which happened in early 2020. And we're also planning on having a workshop this year, later in the fall, hopefully face-to-face, but if not, virtual with FDA. And we also have established a strategy on engaging with regulators outside of the United States. We believe, again, as I mentioned before, right, that engaging with regulators at these earlier stages is imperative to advance this field with these technologies. And we have received an awesome response from FDA. The interaction has been incredible there, and back and forth discussions. We continue to have frequent teleconferences between these engagement. So we really believe that, again, this collaborative interaction will help us understand these technologies and move these technologies and advance them. So what are our deliverables? So far, as I mentioned, in February 2020, we had a joint workshop with FDA and iqmps affiliate, and this was at FDA location. We also agreed to develop the post-workshop publication where we will discuss the outcomes or what happened at the workshop and our perspective on these technologies. And we also worked with FDA to provide us with questions which we could incorporate into the survey 2.0. So they had a chance to look at, and we actually presented some of the survey results at this workshop that we had with FDA. And now having incorporated some of the questions that were shared with us through FDA, it just going to really help us better understand what they are looking for in this survey. And then we also have initiated webinar series. So between these workshops, the face-to-face workshops, we really want to continue our engagement. So we had so far 2 webinars where you have representatives from iqmps affiliate and FDA and addressing various topics and presentations, but we do spend quite a bit of time with Q&A and discussions, which have been very productive. So as I mentioned, we have a communication and external organization outreach group, and it's imperative for us to have this group because, one, to communicate all of the things that we're doing and having a comprehensive communication strategy, even just within the IQ, but obviously also externally. And how to engage with non-regulatory stakeholders, right? So as the field is expanding and growing, there's a lot of other organizations that are engaging or working in this area. So again, we don't want to duplicate efforts. So this group is looking at other stakeholders and engaging with them to see, can we collaborate together or share information. So some of the deliverables, this group has launched an awesome website, iqmpsaffiliate.org. Again, that's where you can find information about us, but also different projects that we're working on, and also having access to the publications that I mentioned earlier. This group has also implemented a social media strategy. So for example, you can see us on LinkedIn. and you can join us there and see updates from this group. One of the things that, as an iqmps affiliate, very often we're asked to participate and present at different events, conferences, or workshops. So each time we had to develop PowerPoint slides or presentations for each one of those. This was pretty intense process. So this group helped us develop presentation materials which we can utilize to communicate our accomplishments and our efforts. And obviously, a result from that is what you're seeing even today that I'm presenting. And we also want to raise awareness with the key external stakeholders regarding us as an iqmps affiliate, but even more importantly about these technologies and where pharmaceutical industry sees these technologies moving forward. So our project proposals team is also very busy and certainly a very excited team. So there's 3 areas here where this project team is working on. So one, looking at ability to share data, retrospective data. So various members of iqmps affiliate, and obviously have been working with these technologies. So there's an opportunity for us in a pre-competitive environment to share the data, right, and where to share it and how to do it, who would have access to it. And as you can imagine, that's not an easy proposition, but we are certainly working on it. And the goal for this specific project is to develop a mechanism and a framework to share, collate, and analyze existing MPS data. The milestones here, we have initiated a call for participants to join this discussion, and we are focusing on specific organs or contexts of use. The other objective here is for joint characterization, right? So identify a specific organ and be able to characterize it. So here we have actually made quite a bit of progress. I shouldn't say actually, we are making quite a bit of progress. The first area of interest was a GI MPS, and then second, kidney MPS. And you can actually go on our website and see an RFI and RFP for the GI MPS, which enable prospective and collaborative projects to characterize these 2 platforms, the GI and the kidney one. The kidney one, we're also have engaged with the PSTC, Kidney Biomarkers team. And again, this came through that other working group, the collaborative group that I mentioned before. The suggestion from there were engaged with the PSTC, and the way identified this is one area where we could collaborate. So we're not duplicating efforts. And then the third project is or category is prospective collaborations. So here the goal is to identify collaboration and alignment opportunities for ongoing projects within our individual companies. So we have initiated a call for participants to join these groups and discussions, and we're going to be focusing on specific organs and contexts of use. We have not defined those, which ones are prioritized those yet, but we're getting there. So the other big question really comes up when we think about these technologies is which species. So should we engage with human cell-based MPS or animal cell-based MPS? And a lot of efforts until now have really concentrated on a human cell-based MPS. And of course, when you think about it, it does make sense, right? We're trying to translate our medicines from preclinical to clinics. We want to make sure that those medicines are safe and effective for our patients. We're trying to do it at a faster pace if at all possible. So working with human cell-based MPS for human medicines makes sense. But there's certainly some areas where the animal cell-based MPS can be helpful. And so here, obviously, I'm going to address in the context of drug discovery and development, but there are also other areas. There was just a workshop sponsored by National Academy of Sciences where animal cell-based technologies were discussed in the context of, for example, veterinary medicine or agrochemical industry. So there are certainly other areas where these technologies are also playing a significant role. But so let's step back and talk about these two aspects within drug discovery and development. So we have come together and have developed a position statement on utilization of animal cell-based microphysiological systems. And here I'll just mention them as animal MPS. So we certainly are recognizing the importance and the impact and the need for animal models. We certainly would not be able to provide all of the medicines that we're currently are providing to our patients without in vivo models. But there are certainly some areas where we can refine where there's, for example, lack of those models, or the models are not as helpful as they potentially could be. So we are recognizing, there's a new approach, new methodologies that we could that could enhance this drug discovery and development path. Exclusive utilization of human cell-based systems, that's the direction we're going to go, and it makes it really challenging to support the investigation of species-specific mechanisms, demonstrate the models, recapitulate histological findings where we have significant amount of data from our preclinical in vivo studies, and validate predictivity for toxicities that only occur above relevant clinical exposures in humans, right? So there's only up to a certain level that we can expose folks that are part of clinical trials. So if we want to go beyond that, obviously we're not able to do that. So animal MPS systems could help us in this specific area. So this is where, again, we're suggesting that MPS composed of cells from other species can help us advance microphysiological systems and their use. So with that, with supporting utilization of animal cell-based systems, where else can they help us, right? So for example, cross-referencing animal cell-based MPS data with enormous amount of in vivo animal data that we already have to accelerate the development and validation of novel MPS technologies, right? We have human cell data, but not certainly to the extent that we have in vivo, the amount that we have of in vivo animal data. We can also leverage animal in vivo data with animal MPS data to gain confidence in predictivity of human microphysiological systems, and also identifying species-specific differences for selection of translating models through MPS species comparison, and especially for the rare diseases. Developing transgenic and disease-specific MPS models with cells derived from transgenic mice and rats. As we know, the transgenic in vivo models have been enormously helpful in evolving and developing various medicines for our patients. So here, utilizing the cells, transgenic or genetically modified cells, can be very helpful. And carrying out structure-activity relationship, or SAR, and mechanistic studies in instances where there are non-clinical toxicology issues that need resolution. So for example, nephrotoxicity or central nervous system toxicity. And then also comparing ADME in non-clinical species and human. So iqmps affiliate supports the 3 R's, the refinement, reduction, and replacement. And I know many of you are aware of these concepts, but for those that are not, those are the concepts that were described in the 1959 by Russell and Birch in the Principles of Humane Experimental Technique. And the current use of MPS technologies has already direct impact on refinement and reduction in drug discovery and development, with the long-term goal of animal replacement in the future. We're certainly not there yet, and it's going to take some time, but that's the potential that these technologies have. So again, I want to acknowledge, right, that here I'm just representing iqmps affiliate. I'm giving the presentation on behalf of that consortia, that affiliate. And this presentation involved a lot of different folks pulling it together. I certainly would like to thank the Secretariat, so Jillian Brady, Catherine Gravlin, Reed Abrahamson, and Mary Devlin Capizzi, and all of the iqmps affiliate participating companies, which I can't spend the next couple of hours naming everybody there. But again, everybody that has contributed to all of the effort and evolution of this presentation. If you have any questions, you can also reach me at the LinkedIn link that you have there on the bottom. But you can also reach out to our Secretariat colleagues, which are listed there, Mary and Jillian. So thank you so much for attending this session. I hope you find it useful, and I'm looking forward to engaging with you on questions and answers and a discussion, and even hopefully offline. Thank you so much.